Okay, class, where we are now, we were talking about the muscle triad. So we want to wrap this up fairly quickly. We said that we had these things called T-tubules. The T-tubules are indentations in the skeletal muscle cell membrane that push into the cytoplasm. Here would be an example of a T-tubule. There's the little holes. And flanking each T-tubules are terminal cisternae. Flanking T-tubules are terminal cisternae. Now, in the terminal cisternae is a protein called calcium-binding protein, also called calsequestrin, in these terminal cisternae. That particular protein holds calcium, a lot of it. So it has a high capacity but low affinity. A high capacity but low affinity. It holds a lot but not that tightly. It holds a lot but not that tightly. What you don't see is that there are calcium release channels right in here that you don't see. The calcium release channels come in and assist, and if they open because of voltage coming down here, they will bust calcium out from there, and calcium will go everywhere in the cytoplasm, and then ultimately attach to troponin C when we put it together. Now back here in the sarcoplasmic reticulum is a calcium pumps. And what the calcium pumps do is to constantly suck calcium out of the muscle cell cytoplasm. Suck calcium out of the muscle cell cytoplasm and go into this cisternae. Then the calcium slides forward here because the calsequestrin pulls it. So these pumps are always on. It's a vacuum cleaner, always on, constantly sucking, and then the calcium that's being sucked is pulled forward and stored here because you have this calcium binding protein. All right? So study this ultrastructure. Study this ultrastructure of everything. All right. Now where we are ready now to talk about how a muscle contraction occurs. All right. So what I'm going to do is, this is where you want to go now. Remember that document that I asked you to print out? Okay, here it is. I wrote this in order to help understand this. All right. Prior to studying this document, study my electrophysiology module that you'll see in D2L. And study the anatomy of the skeletal muscle cell, which we just did, but you may want to restudy it. All right, so let's walk through this. It's only 2 pages long. The modern physiological model of skeletal muscle contraction is termed the sliding filament model. The sliding filament model. See, these things slide. The older abandoned model suggested that a muscle cell would crumple up, twist up, and produce the contractile shortening. Now we know that is not the case. Myofilaments, which you see here, simply slide over one another. The actin slides over the myosin because the myosin pulls it over it in a linear pattern, linear straight line manner, thus shortening the muscle as it contracts. Below is how the sliding filament model works. Okay? So we're going to walk through this, but if you study this first, it's going to make this very simple, particularly the electrophys. All right. So I'm going to say, start here. I'm going to say that right here is, and if you study this, you'll know what I'm talking about. This right here is the neuromuscular junction. This right here is the neuromuscular junction. This is a terminal of a neuron. That's why it's important to study this module. Synapsing on the muscle. Synapsing on the muscle. This is called a motor end plate. Neurotransmitter substance, which is acetylcholine, will come down, and we're going to go through it, but this is my first run through. Neurotransmitter substance, which is acetylcholine, is going to come from these synaptic vesicles, hook to receptors. Once it hooks to receptors, it creates a motor end plate potential. The motor end plate potential is then capable of starting action potential in voltage-dependent gates. Again, all that's in this module. Then what will happen is the action potentials will travel down all away from this site. The action potentials then will go up into the T tubules. In the T tubules, the action potentials will cause the release of calcium from the terminal cisternae. Remember, it holds a lot of it, but not that tightly. The calcium released from the terminal cisternae will go into the muscle cytoplasm. But some is going to hook to troponin C. When it hooks to troponin C, what it will do is to shift the troponin so that tropomyosin comes off of the myosin binding sites on actin. The myosin binding sites on actin. Now, let me show you what I mean by that. I'm going to go back here. See, when we showed this, here was that tropomyosin. Tropomyosin in the resting state covers the myosin binding sites on actin. To get a contraction, this ribbon must come off. What causes it to come off is calcium. When calcium bonds, it shifts the troponin complex and pulls this ribbon off. When that ribbon gets pulled off, then this site is opened up to be able to bond. Okay, so we go back again. So that's what that does. Now the action potentials when it comes to myosin cause the ATPase on the myosin head to split. When that occurs, the energy cocks the hammer back of those myosin heads. Let's go here. It cocks the hammer back of those myosin heads, flips it back. Then what will happen when the heads are cocked back? Also, you open up the actin binding site on the myosin head. Remember, on the myosin head, you also had actin binding sites, we said, on that myosin head. Okay? When that occurs, so here's that actin binding site. When that occurs, then myosin can bind to actin because the ribbon has been removed and actin can bind to myosin, and then the cross bridges flip back and pull it in. So that's essentially the steps in a nutshell. All right. So now what we're going to do with that, we're going to walk through this step by step and see how it occurs. What I'm going to do is I want to start fresh. So I'm going to close this one because that gave the overview, and then we're going to do the excitation contraction coupling. Okay.